My name is Jeff Elleraas. I am a principal scientist here at Pfizer's Global Research and Development, specifically the Purification Lab in La Jolla, California. The big picture, the overall mission of Pfizer in La Jolla is to overcome the toughest challenges in cancer care by extending patient survival, hopefully improving the quality of life for patients around the world. The small goal as a purification lab within medicinal chemistry is to discover and deliver novel treatments in the fastest and most efficient way possible. SFC is supercritical fluid chromatography, and the benefits of SFC is it uses compressed CO2 as a liquid solvent. So you eliminate the need for hydrocarbon, hexane, heptane type solvents, and along with characteristics of being a supercritical fluid, its viscosity and diffusivity within a stationary phase, aka a column, is orders of magnitude better and faster. So you can run it 4 to 5 times faster than you can traditional HPLC. And then some of the benefits of CO2 being recaptured from a manufacturing process. It's technically recycled already. And then you eliminate the need to dry, difficult to dry solvents, aka water, hydrocarbons, that sort of thing. In Pfizer, we are looking at future ways to get the job done using less resources. One of the ways we're doing that is we're looking at smaller scale equipment, aka we are looking at a nano scale HPLC that utilizes, sub-micron particles and has typical flow rates, solvent consumptions at the rate of a fraction of percent of current modern HPLCs. Factor in a smaller mechanical footprint, uses less electricity and less overall resources overall, hopefully to drive us into a more greener, sustainable future operation. So where do I get satisfaction from? Well, aside from working within the oncology research area, and no joking aside, curing cancer, what gives me satisfaction as a chemist, and specifically as a chromatographer, is being able to isolate a specific product from a matrix and being able to separate isomers at a large scale, being able to deliver on something that a normal chemist can't do themselves. AI, and probably more appropriate machine learning, is being used and being looked at to model current chiral separations in terms of how compounds interact with the chiral matrix. We have done some kind of sample collation to see if we can model based on certain characteristics, polar surface area, where the stereochemistry is, what types of groups are functionally around it, and model that with historical 10, column screening data. We acquire data on just about every chiral sample we get, and then we feed that into a machine learning tool that records what works and what doesn't, with the hope that that will eventually predict what will work, so we can kind of streamline the future screening protocols to, hey, this compound should work on column A, B, and C, and then not have to worry about the rest of them. The key challenges facing drug discovery in general are, it's a difficult environment. Within our level, within early discovery, 9 out of 10 candidates that we send into phase trials fail within the first trial. So you're looking at a 10% chance of anything we move forward even getting to the second round. You factor that into a roughly 12 to 14 year discovery cycle from invention to FDA approval. It's a long, arduous task to get things through. Couple that into environmental factors in terms of solvent consumption and just physical matter and trying to get greener and leaner and faster. It's a challenge, and we need to continue to develop these new technologies to work faster, smarter, and smaller, quite frankly.